This afternoon, our lecture is from Dr. Rafael Carrascosa Marzo. He's from Spain. He's recently completed a doctorate in the physics department at Oxford on the topic of designing and constructing complex architectures in the nanoscale using DNA strands as building blocks. So DNA origami. He was working under Professor Andrew Turbofield, who is one of the world's leaders in this field. So you're very lucky today to have the chance to hear about this exciting technology from Dr. Carrascosa. Okay, without further ado, let's welcome him.
Thank you, Scott, for that very kind introduction. And thank you, everyone who made time in their, I'm sure, busy days to come and hear me talk a little bit about this very specific topic. Although I would like to start by also thanking the staff at SCIE, for the opportunity to come and talk to you about this specific science, and also to the CCIR staff, which through these public lectures aims to bring these very specific, sometimes niche academic topics to high school students that are very keen in having a career or learning more about science beyond what is usually in the curriculum in high school. So before I share the presentation and I start with it, I would like to add to a bit of a background so you have an idea where I'm coming from. As Scott said, I'm originally from Spain, where I did my bachelor's in biotechnology, and I later moved on to Denmark to do a master's in nanobiotechnology, later on going to do my biophysics PhD at Oxford University. And throughout this path, I've been very interested in biology, but also how biology can be understood through a physical or engineering lens, and what's the interface between biology and the so-called harder sciences. Because it is in this day and age of fast-paced innovations where multidisciplinarity is the I believe, the correct approach for producing relevant and important research that can bring the most good to humanity as a whole. So without further ado, let me show you the screen. I presume you can see it just fine. So the title of the talk of today is DNA Nanotechnology, from building block of life to engineering tool for small nanoconstructs. So and my current affiliation is University of Oxford and UCL Public Policy, where I'm doing a different line of work. But going a bit from more general ideas to some more smaller concepts, let's say, or more specific concepts. I'm sure you're all well familiar with DNA as a biomolecule that encodes genetic information. That's one of the main roles that DNA plays in nature. It interacts with many proteins and RNAs through transcription, translation, and through post-transcriptional, pre-transcriptional modifications. And as such, it is a key molecule for understanding life. It's not so long ago, or it wasn't so long ago, when the human genome was fully read, not fully understood, of course, but we had access to this information, to this wealth of information. So it is a very well-studied and characterized molecule. But I want to take the talk today to a different direction that you may not have heard before, which is the physical properties of DNA as a biopolymer, as a robust and interesting biopolymer. Because if you think about it, at the end of the day, and you can see here in this schematic, DNA sequence, if we abstract it from this helix, it's just a consecution of somewhat similar nucleotides. So we could consider it, or we could classify it as a polymer, in this case, a biopolymer. But what can we use DNA for that is not, as per usual, this encoding of genetic information? Well, before I go on to that, we need to talk about a concept in physics that occurs at all scales called self-assembly. I don't know if you are all familiar with self-assembly. It's kind of self-evident, perhaps by the name, but it's this property that, again, occurs at all scales. And it leads to the assembly in an autonomous manner or a seemingly autonomous manner of complex materials from usually simpler units. And it's one of the main or most generally used strategies for constructing nano devices, given the difficulties that working at the nanoscale has, since it is such a small scale compared to the regular sizes at the macro scale that we are all familiar with. So using the self-assembly in a directed way can lead to novel and complex new architectures that can have its uses and, most importantly, have the right size for addressing specific topics of interest. So not to try to reinvent the wheel, and just paraphrasing somebody that has a better idea of self-assembly than me, self-assembly refers to the spontaneous formation of organized structures through a stochastic process. Stochastic meaning random or with a source that we are not, we do not understand yet, so it would seem as random. So through these seemingly random processes that involve pre-existing components, that is reversible, that it can be controlled by a proper design of these components, and that is dependent on the environment and a driving force usually coming from this environment. So how does this parse with DNA, the molecule that we are interested in? For starters, what is this unit that conforms the, and I mentioned earlier, conforms the self-assembled structure, these structural particles? Are the nucleotides, and the shape, size, and condition of these components of these nucleotides will define the resulting object or complex architecture. Then what is the binding force between these different particles, these different components, and how do they interact with each other? In this case, it's through hydrogen bonds, which I'm sure most of you are familiar with, but don't worry, we'll point out a couple of examples later down the line. And then something important within self-assembly that requires a certain degree of reversibility or adjustability, meaning it can go backwards, it can be formed and reformed without having to go through any excessive force or excessively harsh conditions. And of course, it exists in an environment, in which case is aqueous solution, given that DNA is a soluble molecule. And then the driving force, what leads to this self-assembly activity, which it is spontaneous because it doesn't require an active input from our side, but it requires temperature and some chemical considerations, chemical polarity. So as I mentioned about the binding forces, there's many different types of forces in nature, as I'm sure you are all aware of. Covalent bonds, van der Waals forces, electrostatic forces in the molecular scale, but we're interested in hydrogen bonds also at this molecular scale. Of course, the bigger you go at the scale, you have other effects to take into account, which kind of emerge from this other molecular scale one. So you have the hydrophobic effects, the entropic effects, electrostatic forces. These particularly hydrophobic effects are somewhat relevant to us, but I won't go into too much detail into today's talk. But just bear in mind that there's these different binding forces at the different scales. The macroscopic ones you will be more familiar with, at least from your own experience, such as magnetic forces. Capillary forces, if you're working with, I guess, solubles like water, for example, and then mechanic forces more directly. But all of these things, of course, do influence in the behavior of DNA in the way that I want to explain it today. But the focus is on DNA. This is an schematic representation of all four nucleotides and how they interact with each other. Thymine, guanine, adenine, cytosine. Thymine and adenine they interact together. Guanine and cytosine interact together as such. And it is these circle dotted lines that I want to draw your attention here, because this is the representation of the hydrogen bond, which ultimately is this glue that binds together the different units of our self-assembled molecule, of our DNA-based construct or architecture. These are very stable, but they can be broken at enough high temperatures. There's other factors to it when we're talking about DNA, but just bear in mind that they come and go quickly, and it is important that, for example, you have only 2 between thymine and adenine and 3 between guanine and cytosine. So having talked about these necessary forces, this, I believe, necessary intro to DNA as a self-assembly molecule, why is it such a good self-assembly molecule? It has the perfect physicochemical characteristics for design and production of complex structures by the relevance of the base complementarity that I mentioned. Thymine will always go with adenine, guanine will always go with cytosine, and it does have some intrinsically great stability because these molecules serve as information storage, so they need to be in their natural application, so they need to be quite resilient for any external effects. Otherwise our own genetic information, our own being would be in danger, let's say. Then, very importantly, which we sometimes forget in science, is that it is cheap to produce. It is with the development of technology, the ease of synthesis is quite great, and we can produce it at a relatively low cost in large quantities. And then, of course, as I mentioned earlier, it's a very well-studied molecule that has a versatile toolkit available for its manipulation. Understand toolkit as different molecules that will interact with DNA and in a very specific controlled way. Think of enzymes, for example, and to a different degree proteins and protein complexes, such as, for example, CRISPR, which some of you may have heard from in the news or we be relatively familiar with, as it was making the headlines not so long ago. And then, of course, as you saw in this schematic, the chemistry is relatively straightforward. So you can conjugate it with other molecules with relative ease and in relatively mild conditions. You don't need any complex solvents or any complex environments to work with DNA, which is a big boon when you're working in the lab with the actual DNA strands. So this kind of introduction was to talk about the underlying concept that drives everything else that is going to come now, which is self-assembly. So I hope you understood that DNA sequences, by virtue of this base complementarity, adenine goes with thymine, guanine goes to cytosine, it's going to bind together in a specific manners and in a very stable way, but not in irreversible way, and in a relatively quick fashion in simple environments. So this is the core of what drives DNA nanotechnology. I will do a bit of kind of look back into what DNA nanotechnology, well, where did DNA nanotechnology come, because it's a bit important to see the historical perspective. But just so that you have an idea, we make use of this base complementarity to create a specific constructs, as the one that you see here or here, which is the same one but in a different state, and to drive changes in the conformation. So, for example, I won't go into too many details of what TET or TAMRA is. It's not very important right now. What I want you to have a look at is the structure. So this, if you look at it, may resemble a tweezer that you would use for plucking hairs. And the idea is that this is in a very, very small scale, and you would have millions or hundreds of millions of them in parallel working together. And the idea is that you have something that is open, and through the introduction of like here, and through the introduction of the right sequence with the right DNA strand with the right sequence, you can close it with this very precise motion that it's going to be easily reproducible and easily reversed. So in this case, just to explain a little bit, the blue color here and the blue color here means that they are complementary, meaning that the sequences are reverse complementarity, because again, I won't go into too many details, but the DNA strands in a helix are anti-parallel, so they go in opposite directions. They don't have symmetry. So they don't have an axis of symmetry. So the blue goes with the blue, the green goes with the green. So basically, by introducing this sequence, manages to put together these two strands. And because this bit here is flexible, as it would be in a tweezer, you can go from open to close in a similar way to this, you could say. And it is reversible, because if you heat up, like this would occur in water at room temperature, but if you heat it up to a certain temperature, say 40 degrees, 50 degrees, this would all fall apart. But it's not a one-trick pony. It can do many other things, or you can take it in different directions. So if you have the right tools. So if you notice, there is this bit here, this orange bit, that hasn't interacted with anything. But if you add a sequence that does have the orange bit, it will be able to attach to this, and through a process called Toll mediated strand displacement, which don't worry, you need to remember this uh, long word. But through this process, you can remove it. And what you achieve by removing it is having this, what we call a waste complex, because this is a fairly inert element in our system that goes to the side and reopens the tweezer. So basically, so that you understand a bit the gravity of what we have achieved here, is we have a system that has one conformation open and then has a different conformation close, and we can cycle through the two of them with very easy, in a very simple step manner, in very mild conditions. And this is relevant because this is all occurring in the nanoscale in very, very, very small sizes. Because just so that you have an idea, a nucleotide in the width, so one of these lines is 0.34 nanometers. So in a millimeter, you have 10 to the power of 9, yeah, 10 to the power of 9 nanometers. So think about the size of a millimeter, which is something that you have more clearly in mind. So if you reduce it, if you divide it in such a great number but in equal sizes, you will have 1 nanometer. And this structure is less than 1 nanometer. Like this, not this whole structure, but each element of this structure. This is a very small scale. And of course, this is just working with DNA, but as I mentioned earlier, the chemistry is quite straightforward. So you can introduce different molecules that are not exactly DNA. In this case, it's an isobenzene, which is a molecule that changes conformation with UV light, visible light. And you can induce the same procedure, this closing, opening of the tweezer, but through a different set of components and through a different trigger, which has its advantages and its disadvantages, as you can imagine. And it's not only just having something that switches between two positions, you can also have something that produces some sort of work. So in this case, and Scott mentioned earlier, my former supervisor, Professor Andrew Tuberfield, and one of his biggest contributions to the field was the production of a DNA walker, which, if you look at this black element as imagine that it's a little person with two legs, these black lines, and then this green bit here with the blue and the yellow would be the shoes. So he developed a system with the people at the lab that could step away from this position and go over, as you see here, in different steps, so that it would step forward in this, I guess, to the representation, step forward towards the right. So by cycling through the introduction of these relevant elements and with this complex interaction of the sequences here, because this is not as straightforward as this tweezer, it is a bit more complex, but I won't go into that detail. By introducing the right elements, you can force the system to change its state and take a step forward. Or I mean, of course, it can go backwards or not take a step at all. So it kind of idle step. It just raises its feet, as you can see here, and then just puts it down in the same place. This, of course, is not the ideal, but it is given the system, the limitations of the system, is the situation that we find ourselves is we can take a step forward, or we just fall back into position. The thing is that by cycling these elements constantly, you achieve kind of a random distribution of steps forwards and backwards. So as I promised, I wanted to talk a bit about where does DNA nanotechnology come from exactly, because it's not an entirely self-evident proposal. This that you see here, this cross-shaped structure, is what we call a holiday junction. So basically you have 4 sequences in black here that are have their base complementarity with 2 other neighboring sequences. So this one here on the left is complementary with the top right and with the bottom left. And it can have different conditions, but basically what you get is this kind of cross, which you can kind of abstract into this more schematic representation. So Ned Seaman was a professor in the US, which sadly recently passed away. But he was in the late 90s, mid to late 90s. He started considering, if I have this cross, which is a very reliable and small kind of junction, and I put several of them together, and then I not only in 2 dimensions, but in a third dimension as well, like achieving this kind of 3D disposition, I should be able to use it as a scaffold, as a place where I can put other things that I can study. So to understand where he was coming from, he was a X-ray crystallographer, which I don't know if you're familiar with the technique, but basically it's a very complicated technique to use for, but very useful to study the structure of proteins. You basically have a protein, you make a crystal out of it, and then you can, through X-ray, you can understand the structure. This is very simply put. The problem is that it's very hard to make a crystal out of a protein. But if you can create the scaffold into which you put your protein, in this case, it would be these little circles here. If you can arrange it in a particular way, then you are effectively creating the crystal that you need in order to use this technology. So initially, and this is what I want you to fully see here, DNA nanotechnology most of all is a tool for other disciplines. It is a very useful tool and a sometimes complicated tool to use, but it is something that can help you understand other things. In this case, structural biology. What is the shape of this protein? What are the different elements of this protein? This man, Professor Ned Seaman, tried to achieve through this kind of scaffolding made out of DNA. Of course, it wasn't very successful because it's very difficult to produce these holiday junctions and create them in that fashion. He did get some good results, but quickly the field evolved outside of this purely X-ray crystallography application idea. And one of the first steps that came into it was trying to add a bit of more complexity to this very simple cross pattern, to this holiday junction, to add more robustness to the whole thing. Because DNA, I mean, you can imagine that it can be a long sequence, but it is very, to put it very prosaically, it can be very wobbly. It can be too flexible for its own good. So if you're trying to do something more rigid, which doesn't really work like that in nature, you need to make it a bit more complex. So in this case, for example, instead of creating a lattice with these crosses, they created a lattice with these tiles, which is only 2 different tiles, A and B. And if we have a more concrete look at this kind of schematic tile, or this section of tiles, this is how it looks in reality. So it is a bit more complicated because here we see only an abstraction, but still the DNA sequences. And in this case, what we would call A would be this section here that I'm trying to point out with this pointer. And then the connectors would be these yellow lines that are not fully inside the structure. And then B would be this bit here and this bit here. And then A would be this bit here and this bit here. So as you see, this isn't as simple as a cross of DNA, a holiday junction. Now we have 1, 2, 3, 4, 5, 6 helices if you look at them this way. But the helices don't just go as you have seen it in the previous, at the very first schematic I show you, or in your lessons, that they just go with as a single helix going in this way. As you see, the DNA helix is formed by red, for example, is coming along the sequence, along the strand, but then it crosses over to the neighboring helix and then goes back down. So these are interconnected helices, and this acts as a bit of a glue between all of them. And that's kind of the basis for preventing these things to be too wobbly or too unstable. You kind of weave them all together into this tile that then interacts with other tiles that through this further weaving of these different sequences, and of course all based on the sequence, like the base complementarity of the sequences that I was talking about earlier. Remember the thymine with adenine, guanine with cytosine. So there's many different ways that many different shapes that DNA nanotechnology can take, and they are all very useful, and they are still being developed to this day. But the one that I have worked with in the past, and the one that has been driving more strongly these bigger structures or more complex structures, is DNA origami. But before I go to showing some specific examples, I would like you to understand what is the DNA origami, what's the principle of DNA origami. So I'm sure you're all familiar with origami as a papyroflexy technique. You have a piece of paper and you introduce certain folds, and you can go from a simple 2D, mostly not 2D, but almost 2D element, the sheet of paper. You can fold it into a complex structure like a little bird, a crane, and so on. So with that idea in mind, there's they use that title for the DNA origami because it's a bit of that kind of lines up with what happens with DNA. In this case, our sheet of paper is this long single-stranded DNA, which we call scaffold, and then the folds into this sheet of paper are induced by these short oligonucleotides. Oligonucleotide is just a very short DNA sequence that we call staple. So basically, if you look at it, this purple region and this green region, they are very far apart. But by base complementarity, if we introduce the right sequence, the purple section will interact with the purple section, as in here, and the green will want to interact with the green section. So all of this is in constant motion, is in constant vibration, because it's very small and it is in water. So I won't go into the specific physics of it, but basically everything is moving around, vibrating. And this allows these two parts to get close enough together to bind and basically create this fold here that wasn't here before. So you're basically taking your paper sheet and doing the first fold through this. So if you start doing many different folds without having to break the paper, without having to break the scaffold, you can produce a defined structure. In this case, it's this square. You can make this pyramid as well. There's a bit more detail in this schematic, but ultimately, the idea is to make things way more complex than that. And in the seminal paper that introduced this concept in 2006, which now is some time away, but is relatively recent for what a scientific field is, they produce a smiley face, which I always found very funny, because they were basically producing lots and lots of very small smiley faces out of DNA, and they could see them directly. I think I have the figure there. But before I go on to that, I want to show more directly how does this work, because it's all well and good to try to explain it, but there's this like a video sometimes it helps much more than that. So you see here, this first big line is the scaffold. The quality is not that good, sorry about that. And then you see the shorter sequences, and as you see, they are all in random places interacting with the bits and straightening things up, and they are kind of falling into place. It is not an ordered process, it's going a bit randomly as the structures and the areas get close together, and basically what you end up is with a square. So from this initial long winding uneven line, you get to this specific square that you saw at the end. And that's kind of like how this DNA origami works. What's the principle of it? Of course, that's just the square, which it is very simple, but I mean, you have to design it. But of course, you can make more complex things. Like, for example, this is the paper that I was referring to. These are all schematic, but these two bottom rows of images are from AFM, which is a specific type of microscope that lets us see things very, very, very small, to almost to the atom. I mean, to the atomic scale. With the right definitions, but the right parameters. But basically, they could design DNA, this sequence, and the interactions in a way that it would fold into a smiley face that you can see here, and not just one, but many of them. Because when you're working with DNA, nanotechnology, DNA origami, you're working in the hundreds of millions of elements occurring at the same time. Because remember, we don't do anything directly to these sequences. We are depending on their intrinsic characteristics, chemical characteristics, and the temperature of the system to actually produce these things. So, and of course, we don't need to be limited to the 2D. You can also produce in 3D. These images are also real images of how it looks at these structures made out of DNA. This is also the same technique as this one, AFM, atomic force microscopy. And then this is something a bit more recent, but with a different technique, which is cryo electron microscopy, which again, these are all different types of microscopy that don't use light because light wouldn't allow us to see something so small. We use different physical concepts, let's say, to be able to visualize these things. So of course, those are at a relatively small size. I think the smiley face was in the 100 nanometer kind of size range, which is not very big. Sometimes you want things to be that small, but sometimes we like to push the boundaries a little bit of the field, and that's what she, which is also a very big, very relevant name in the nanotechnology professorship at Harvard University. He tried to see how big can we make these structures, and through this clever design that I won't go into too many details, just suffice to say that he was using these little blocks of 450 nanometers in size, more or less. By putting them together through this clever design, he could make these huge, comparatively huge structures, 2 micrometers, which is 3 orders of magnitude bigger than the nanometer. So just to say that this is still very, very small, but these things are way bigger now than the single strand that I was talking about. There's hundreds of thousands of strands in these structures over here. And of course, as I said, we don't need to limit ourselves to 2D. We can also look towards 3D. So originally, the structures were very conservative in the size and shape. So the simplest thing that you can do in this case, if you have like these cylinders, is to make a cube of sorts. They also work by instead of having everything rigid, bending things around. So they made this kind of little bottle shape, this tetrahedron. All of these images are with electron microscopy, but this is actual DNA. Like these lines, these white lines that you see here, are the actual DNA helices that we can see with this technology and that we can order in this very controlled fashion, which to me is always amazing to see, because this level of control over something so small, with other types of technologies, other type of materials, is very difficult from a materials point of view and from a skill point of view. And these are just very inert structures. This is something a bit more recent that was done by Professor Dietz at the Munich, which is also one of the most relevant names in the field of DNA and DNA technology. And he was basically, during the whole pandemic, trying to consider how we could use DNA, and in this case DNA origami, for attacking viruses, helping with that. And basically he had these huge DNA origami structures formed from smaller pieces that kind of stack together as if they were Lego. And he remember that you can add the different things to this and make it more interactive, let's say. He added the specific antibodies or fragments of antibodies that could recognize the virus. And basically what the idea was to use these little DNA leads to block virus molecule in place and not allow it, like reduce its infectivity and prevent it from propagating the disease. That was kind of like the idea of using DNA origami in this scenario. But what I personally like, and is what I will be talking about a bit more now, is the dynamic DNA origami. So far, we've only been seeing static structures that either are the surface in which other things will be added and will be acting, or are just static squares or these static leads that do have one function, but they do not change conformation. what was the basis of my work and what I particularly like is when it has this change of conformation. Like, remember the tweezers that were opening and closing? This is that same idea, but taken into the bigger scale, let's say. So there's been a lot of work on it in the past decade or so, kind of making these different structures, as you see here, imitating engineering concepts that we see in the macro scales. So, for example, these levers that move with respect to each other, and it's again made out of DNA, or this cylinder that has this slider going along it, or a combination of both, so that you have basically like this sort of piston structure that you could use to propagate movement if the microscale rules of physics applied in the same way in the nanoscale. Something very interesting as well were rotors, structures that go in circles, and they are able to go in circles either randomly, because remember, thermal fluctuations is shaking everything. So if you're clever with your design, you can make structures that rotate mostly in one direction, so you create this kind of coherent movement. Or you can use external inputs, like for example electric fields. I didn't mention this before, but DNA is a negatively charged molecule. So if you use cleverly these different electric fields and you kind of change them or turn them on, turn them off in a specific way, you can make a DNA arm, which is this thing, this blue thing here, rotate and orient itself in a specific way, and you can make the arm move at very, very high speeds. And then, of course, you have things that have more clear application, because so far I've been talking a lot about what we can do, but not to why would we do it, or what would be the application, which is fine in science. It's important to have our basic research and explore these different things, but it's also nice to find real world applications for our work. So in this type of structures that you see here, one specific molecule will attach to a place and lead this changing conformation. So in this case, you have this kind of open structure, but once you have this cargo molecule in place, it will close it down. So this change in structure basically is telling you, in the conformation is telling you that this molecule is there. And the same goes for here, from an open X structure to a closed kind of I structure. These two structures could work as biosensors, telling you be careful because you have this molecule here. So it could be a marker for a disease, it could be a contaminant in wastewater. And the good thing is that you only need one molecule to lead to the change in conformation, which means that you have a terribly high resolution compared to other types of biosensors that you could find around. It's not the most precise one it's not necessarily the one with the greatest resolution, but it does have a very high resolution. And in a different way, in this case, you have these little boxes that basically, without going into too much detail, you can add something that makes the box open or close. So if, for example, imagine that these pink things here, in reality, these are nanobodies, which are fragments of antibodies that have a therapeutic property. But just to explain it more simply, these pink things, are these proteins, have a therapeutic activity. They do treat an ailment, a disease. So as often happens with complicated diseases, the cure may be worse than the disease, or the cure in the wrong place may be worse than the disease. So most of what nanomedicine is about, which is a field that I worked before going into DNA nanotechnology, is about encapsulating these molecules and preventing them from making more harm than good in the rest of the body. Think about chemotherapy, which sadly is a very aggressive treatment for the patient. So if we can put these chemotherapy drugs inside a box made out of DNA, and this box of DNA only opens in the right place, then we have improved substantially the treatment. And we can modify DNA so that it survives in the bloodstream. It doesn't survive that well on its own, but we need to add different components, but it is easy to add these components. And we can make so that the opening of this box, like the lock that locks this box, only opens with a certain key with a certain molecule or structure that is only found in a specific place of the body, thus ensuring the specificity of the delivery of this drug. So it has these applications in pharmaceutical endeavors in that sense, pharmacological endeavors, or nanomedicine in this case in particular. But it's not only that, of course, we are also looking at templated chemistry, in which you basically have a long chemical reaction, but all the components are one after another in this DNA surface, and we can easily control the distance and the number of these different components. So they basically are like, you could consider them as dominoes that you start on one side and they fall into place and continue the process further down the line. Molecular electronics is quite interesting, which is using DNA and other kind of molecules for making transistors and different elements of electronical devices. Because remember, DNA can have, I mean, does have a negative charge, so it can have current running through it without too much difficulty. And then, as I mentioned earlier, with this changing in conformation depending on the presence of certain molecules, it can be used in biochemical research for studying the molecules that are in a system, for producing biosensors, and so on. So all of this is well and good. I have talked in very general terms about DNA nanotechnology. I've tried to explain a little bit what drives the behavior of DNA nanotechnology, and more specifically DNA origami. But I wanted now, towards the end, to talk a little bit about my own research. I work with Dr. Eric Benson and Dr. John Bath at Oxford University on producing what we were attempting to be a printing mechanism in the nanoscale. We have a couple of publications out on these concepts, but basically the idea is, think of your regular printer, the one that you use for printing papers or things like that. Imagine if you could have something like that, but in the nanoscale, again, in a very, 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 very small scale. And instead of printing sheets of paper, you would print materials, for example. There's a limit in the production of these very small features in materials or in smart materials. It is very, again, labor intensive and very costly. So our proposal was, if could we make this structure out of DNA, and we make hundreds of millions of them and have them work in parallel over a surface, and introduce changes in the surface with incredible resolution, with atomically precise manufacturing is the idea, so that we can move atoms around, or we can add one molecule in one specific place at a given time, things like that. That's kind of the rationale. And how can we achieve this level of control in such a small scale? Through DNA origami, through DNA nanotechnology. So my colleagues and I developed these two different structures, where this kind of quarter of a circle with this line here, and this white line would move along this arc here, and the red bit would move along the arm of this white arm, rail we call it, and we would have polar positioning. So you would have a greater density of positions here and a lower density here. And then something more traditional, which is Cartesian positioning devices. So just think of your regular two axes. This white arm moves along this y axis, and the red circle moves along this arm again in the x axis, and you basically have a homogeneous cover of this square area here. So that's the idea. Then how does it look in DNA? Like this. This is not actual DNA. This is from a simulation, a coarse-grained simulation, but it is a very close representation of how we would look in DNA. So if you look at it, we have all the different DNA helices that are running throughout this structure and kind of putting it together. And they are, in this case, for example, we have two components. The red would be one and the yellow would be a different one. And in the Cartesian positioning device, we have three. The canvas, where we're going to be printing, the green bit, which are the arms, and then the yellow, which is the header of the printer, no, that is moving around. So to look at the one that I specifically work with, this polar positioning device, I call it the bow and arrow device because to me it looked like a bow with a knocked arrow and pulled back. This I haven't gone into how do we actually do this because if you think about it, this structure, for example, has 8,000 nucleotides and all of them have to be in the right place and interact with the right nucleotide. So it gets quite complex really quickly, but I don't need to do this by hand, thankfully. We have a specific software for this that I haven't explained here, but basically this is how it looks in the software. This is again a schematized version, but you can see how the line goes this way and then comes through and then basically runs throughout this whole arm and then comes back out and then goes to the other arm. So as you see, the routing is not as straightforward as just like a square. The more complex your structure, the more complex is going to be necessarily the routing, the disposition of this scaffold. But there's ways of being clever with this and producing these complex dynamic structures. So ultimately, this is a figure from the paper. This is how it looked. Like these are the different elements on its own. With electron microscopy. It is a bit hard to see because they are kind of each on their own and they are flopping around a little bit. In here you can see though, like the bow and arrow with the arrow, this little arm in position. But in here you can see on the other side the whole structure assembled. So in here you can clearly see this Cartesian device system in which you have the arms, see, in green, and then this black dot here would be the yellow bit, and then the lighter gray bit is the canvas underneath. And the same goes for the bow and arrow. And then we have not only this type of microscopy, but other type of microscopy with, this is super resolution microscopy technique with using fluorescence. Again, I'm not going to go into too much detail about it, but basically it allows us to use light fluorescence to see these very small structures, which is not an easy fit, but you get these very clear images in which in red you have the structure, let's say the printer, and in blue you have this, what is yellow here, this little blob here. And this whole led to using these devices for patterning a surface. So we could, in the case of I'm going to show the results of the Cartesian device, we could place the right head, this yellow bit, what is actually going to do the changes on the canvas. We could place it in 9 different positions with relative ease. In blue, you see here in light blue where more or less it falls. Like these images are an average, so it's not exactly clear here, but they are in the position that we ask it to be with as by introducing specific sequences. And then with this, again, I won't explain this into too much detail, but with this complex mechanism, we could print ink in the canvas. So the ink would be these fluorophores, these elements that emit light, and they would only appear wherever we have our right head, this yellow element here, which is green here. So we put the green element here, and then it prints in pink here. And we could do up to 2 steps because this is just one step. Now we put the header here and then we print, but we could make it so that we print in one corner first here in the bottom left, and then we print in the top right. So this was the level of, and these sizes are under 100 nanometers. So we are able to put 2 very defined components into 2 very specific positions in a very small scale, with great resolution, and at a very low cost, because we don't need very difficult machines to operate. We don't need very hard conditions. We don't need vacuum. We don't need high or low temperatures. This is at room temperature, which is for us it is a great fit of where the science has gone. And that's basically it. That's the end of the presentation. I hope you found it interesting. I tried to make it as wide and encompassing as I could. Before we move into questions, I would like to mention that this is a bit of a primer of what the course that I give at the CCIR Institute is. In this course, we would go into greater detail about the molecule of DNA, the physics behind it, and how do we actually do it. And the best part is that you could actually do your own structures, because most of these tools you can find online. So with our guidance, you can make your own little square, your own little box, things like that. And if you're not very keen on that thing in particular, there's many other different resources and courses on coding and something more biological, less at the interface, depend on what your interests are. But that is it. Thank you very much for the opportunity of talking here today. And I'm open to questions now if there would be any.
We have time now for a few questions. Who would like to begin? Okay, I'm going to start with a question then. I was fascinated by the DNA walker that you showed us from Professor Turberfield's work. It puts me in mind of the kinesin walk, right, along microtubules. I wonder if you could comment on the extent to which these DNA structures could mimic proteins, and maybe to the extent that we could imagine life forms which are made purely of DNA or RNA. Is that feasible?
Thank you. That's a very good question, because it is, as you say, it is the inspiration for these walkers is how these kinesins, these myosins walk around these tubules in the cell. The issue with this, though, is that these structures made out of protein have certain chemical, let's say, characteristics, and they are quite complex systems, so they are able to do this function quite fast. Like, they walk quite fast if you compare it with a DNA walker, and they do have this very well-defined directionality. What I showed in that slide is one of the, not the first, but the first that Professor Turberfield did, but one of the earliest examples of the linear walkers. By adding different elements that are not only DNA, we could make it so that it would certainly only walk in one direction. We could try to make it go a bit faster, but it is still a very rudimentary kind of imitation of life, let's say. So DNA on its own, this has been really looked into. It's not really, I mean, the way that I see it, it's not really a material that will be able to work on its own to create life or create a complex mechanism as our cells, but it is an integral component for such synthetic, let's say, cells. We would need the DNA to play the role of the scaffold and to play certain roles, but we would also need other elements like proteins to do what proteins do best, which could be this displacement in one direction or catalyzing a certain reaction and so on.
More questions?
So good morning, good afternoon, Mr. Dr. Marzo. So after listening to your presentation about the nanotech and DNA, I couldn't help but wonder if there's any like real life example where doctors and scientists use these knowledges to make human life more convenient and easier.
Sorry, so the question is about the applications to make like life easier. Was that the question?
Yes, real life applications.
Yeah, so as I mentioned earlier, most of this, like the smiley face and these 3D structures, they do not have an intrinsic application to it, which is not necessarily bad, because this was produced when this was a bit of a playground for trying different things for them, and like seeing what were the limits or the scope of this field. But basically, what has been used for in the past is for, as I mentioned, the encapsulation of drugs with pharmacological application. So you can use it as you would use other materials for enclose this drug that is very dangerous for the rest of the body, so that it only opens in a specific place. You can use it for like this viral entrapping. This is quite new, because this is only from last year, and so there's a lot of caveats to it, but you could consider it as a different strategy for blocking out bacteria or viruses. You could use it as parts of transistors. I wouldn't make a transistor only out of DNA, because it has been tried in the past and it doesn't work that well. But if we are trying to move into electronics and away from materials that are a bit limited in their availability, DNA could be such an element. And something quite interesting that was being looked at that is not exclusively part of the nanotechnology is using DNA as a storage for computers. So storing data and information in basically you convert the information from binary into quaternary into the nucleotides. And basically, if you have a sequence with a specific, a strand with a specific sequence, you are encoding a specific information. So it can be used for computers, for example, and simplify data storage.
Okay, more questions. Okay, that brings our lecture today to an end. Please let us thank once again our guest lecturer, Dr. Carrascosa.
Thank you.